Welcome, everybody. I'm very happy to start the seventh webinar of the online edition of the Protein Electrostatic Conference. Today, we will have our first fresh talk. And so today, we will have Artemi Bendandi, who is a PhD student from both IIT and University of Genoa. And then we will have a talk from Alexei Onufried, which I will introduce later on. So the title of the presentation of Artemi will be Modeling Electrostatic Interaction and Solvation in Chromatin. So Artemi, please, you can start.
So, hi, everyone. As Walter said, I am Martin Bendandi. I am just concluding my PhD in physics in IIT and in the University of Genoa. And actually, Walter is my supervisor on my project. And today I'm going to be discussing mostly about what my PhD thesis was about. And yeah, so thanks for having me. It's really nice to be able to show my face around here. So, okay. Yeah, okay. So what is chromatin and why are we interested in studying it? So chromatin is, as probably most of you know, a molecule in which DNA binds around some proteins named histones. And in these, around these, oh dear. Okay, let's take it from the top. Okay, so chromatin is a molecule in which DNA is wrapped around some proteins that are named histones, and it forms spool-like structures. So you have these spools that are then connected by linker DNA. So this happens because otherwise DNA would be unable to compact itself, and the tuning of gene expression, like the accessibility of DNA to transcription factors, would be very difficult. So why are we interested in studying this molecule? It's because we know the structure of nucleosomes of the basic entity at atomic resolution, but we really don't know what are the determinants of the second level of folding of its topology. So we want to understand what does chromatin look like and why. So of course, this is an electrostatics meeting, and we're going to be discussing electrostatics in chromatin. So electrostatics is very important in chromatin, most of all because DNA is very highly charged. So in order for it to be able to wrap around proteins and to compact itself, the self-repulsion between the gyres of DNA needs to be balanced, and this happens with the interactions with piston proteins and sulfate ions. So this leads to electrostatic stabilization of the fiber. Of course, as you can understand, for such a charged molecule in such close interactions, electrostatics emerges as a key determinant of chromatin remodeling, of what is going to be the topology of chromatin. And I hope I'm going to convince you with this talk that modeling electrostatic interactions between nucleosomes and in nucleosomes with reductionistic potentials without taking into account the role of the histone tails, of counterions and solvation, and of nucleosome positioning does not yield very accurate results. So first of all, what did we look at? We started looking at the interactions in nucleosomes, electrostatic interactions in a single nucleosome, and we wanted to study the importance of the histone tails. In order to do that, we developed a methodology to associate conformational changes in protein DNA systems with their electrostatic effects. And this methodology comprises of the following basic steps. So in the beginning, we performed some clustering analysis on a molecular dynamics trajectory, either that we have or that we can obtain, in order to extract representative structures. This gives us static structures, but which at the same time reflect the dynamics of the system. So we look at several, let's say, snapshots of what is happening. Then we calculate the electrostatic potential and the electrostatic field on the DNA on each of those structures. And we are able to infer the electrostatic forces acting on the DNA, and then we compare those to a protein-DNA contact map. Basically, we want to see how the field, the potential, and the forces change when we have a different number of protein-DNA contacts, so protein atoms near the DNA with thresholds that we can choose. Finally, we correlate the structural characteristics that we see to electrostatic effects, such as the role of specific residues that we see maybe are causing some particular spike in the electric field or something like that, the role of intrinsically disordered proteins, as we did here for the histone tails, the truncation of terminal regions, and such. So, as I said, we applied this methodology specifically to the histone tails. The histone tails are the intrinsically disordered terminal regions of the histone proteins. We have the main histone octamer around which the DNA is spooled in the nucleosome, and each of those 8 histones has one N-terminal, and 2 of them have also C-terminals, so a total of 10 tails. And these tails are rich in positively charged residues. So we applied our methodology to these structures, and what was most striking is that we saw a dependence on histone tail positioning on transcription. We were able to separate the effect of the radial and the axial electrostatic forces acting on the DNA gyres in the nucleosome, and we were also able to study the effect of histone tail truncation. Among else, we also studied the effect of particular arginine and lysine residues in the nucleosome, and what small changes in their positioning, what their effect was in the electrostatic interactions in the nucleosome. And something that I also really liked about this work is that we showed actually that the C-terminal tails are important as well. They are shorter than the N-terminals, and very often, sadly, they are not taken into account, but we showed that small changes in their positioning gave rise to large changes in the electric field and therefore in the interaction. So besides looking at electrostatic interactions in nucleosomes, we also looked at interactions between nucleosomes. So what did we do? We used Delphi. Oh, and in the work before, we used Delphi as well. We used Delphi interface with NanoShaper, which is something I'm going to mention a bit later on as well, on structures generated through Python scripts for intermediate distances or using the Haddock software for very close distances in order to estimate the interaction energy between nucleosome pairs. This led to a code submission in BioPython. So we developed a parser for PQR files and then input-output module for PQR files. So those are part of BioPython now, which is cool. And yeah, so we started from the 1KX5 crystal structure, and we sampled the possible rotations and translations, therefore forming different nucleosome pairs, and then recorded the electrostatic energy of each possible combination. We also studied the influence of the presence of linker DNA by doing basically the same thing with structures that had linker DNA in them. And we are also interested in the relative orientation of nucleosomes, because the different relative orientations may give rise to feasible or unfeasible sterically structures. And this has not only a local effect, but also it can have global consequences. Because, for example, even if the configuration is feasible, it may give rise to different linker DNA positioning, which then gives rise to a solenoid or a zigzag, or it can have repercussions on the start of the fiber and therefore on the topology of the fiber. So, as I mentioned, we used NanoShaper. We used it to apply the steric clash criterion. So basically, we calculated the volume of the structures, and if there were clashes, we discarded those. This led to less statistics for close distances, as you can see here in this plot, but it also gave us a sort of a negative result. It told us which are the non-feasible angles and pairs of angles and of angles and translations in very tight packing situations. So yeah, so we were also able to determine a threshold after which the monopole approximation is valid. So we saw that basically the energy started following the monopole approximation, and we want to use this data to parameterize a functional form in order to be able to describe what electrostatics looks like in chromatin, basically. So this is why we wanted to create this map of interaction energies between nucleosomes. So here in this plot, you may see some points that look like outliers, and that's actually what we thought in the beginning, but actually it turns out that these situations, these configurations that that show a different energy than what we were expecting for their given position, actually those are particular configurations of linker DNA. So we saw that maybe the linker DNA configuration was like this, or maybe it was like this. And we think that's cool because it shows that linker DNA positioning is also important, not just the position of nucleosomes themselves. So yeah, we said what we are doing for close distancings, for intermediate distances, but where do we want to take this? So for large distances, we're just looking at the monopole approximation. The nucleosomes are represented as the electric spheres immersed in a solution with just the monopole charge placed in them. For intermediate distances, we also have the emergence of a dipole inside the nucleosome. This introduces the orientation dependence, which, as we saw, becomes relevant. And what is nice is that for large distances, this naturally falls into the previous case because the dipole that term becomes less and less important. And since we have numerical data, we can also estimate how feasible this approximation is. So finally, where this project is going to go, even though I'm finishing my PhD, in the future, what's going to happen is that we want to develop a custom force field for chromatin, a custom ultra coarse grained force field for chromatin. So we want to expand everything into 5 interacting centers. One is going to be placed in the center of mass of the nucleosome. 2 of them in the linker DNA entry and exit sites, because this gives us the orientation dependence, basically, and 2 more on the linker DNA, so as to be able to see if the linker DNA is straight, if it's curved, or generally what it's doing, because as I said before, this has repercussions then on the global topology of the structure. So something else that I mentioned at the very beginning, besides linker DNA, besides just treating nucleosomes as spheres, I said that I would try to convince you on the importance of solvation in this system. So what we did is that using NanoShaper, we looked at the porosity of the nucleosome. We looked at its surface area to volume ratio, the number of cavities, the number of pockets it has, and we saw that it presents high solvent and dissolved ion accessibility. And this, of course, would enhance screening of the charges between the charges of the histones of the DNA. And we performed a quantitative description that we found to be in agreement with qualitative data from previous studies. And actually, what is cool is that we were able to, with NanoShaper, to see the channel that traverses the histone core. So in the histone core, there's a central channel that traverses it, and it renders the nucleosome permeable to solvent and therefore to both electrostatically and biologically relevant ions. So regarding NanoShaper, I just wanted to say a couple more things about this software. So NanoShaper, some of you may be familiar with it, some of you maybe not, and I suggest that you become familiar with it. So it is an SES builder, a solvent extruded surface builder that was developed basically for Poisson-Boltzmann applications, and it is interfaced with Delphi. And as you can see, using NanoShaper, we can have more or the same, or like a higher or the same level of accuracy with Delphi without needing to have too large of a scale. So in blue here, you can see an energy calculation done with Delphi with different scales. And in red, you can see the difference in the measurement of the energy when Delphi was interfaced with NanoShaper. So basically, we had very small changes when it was interfaced with NanoShaper, while when it was not, in order to have an accurate prediction of an accurate measurement of the energy, we needed to have very large scale. So this is because NanoShaper provides an analytical notion of the SES, which is independent of grid resolution, and therefore Delphi can extrapolate the polarization charge on those analytical surface patches, providing this more accurate calculation. Here you can see, another thing about NanoShaper is that you can create very cool figures, which is always nice. Here you can see the electrostatic potential map of the SES of the satellite tobacco mosaic virus. And this leads me to my next point, which is the fact that NanoShaper has proven to be very robust, even in cases of very rare systems. You can construct, for example, something that has been done, the SES of the H1N1 viral capsid, which is 14.5 million atoms, and it was absolutely doable. And it is also interfaced with VMD, so you can directly use NanoShaper to represent molecules in VMD, and the benchmark there for a very large system was Clathrin, which is a protein involved in nanocytosis that you can see here on the left. Here, I'm just putting a summary of the main features of NanoShaper. It is rather flexible software. You can do plenty of things with it. You can construct different types of surfaces. You can estimate volume, surface area. You can import different formats of meshes, which is useful if you want to import things that you have already maybe generated. It can you can generate images based on electrostatic features, as I showed before, and it can be freely downloaded from Walter's website. So that's that for NanoShaper. And finally, I wanted to close with some experiments that we actually did for my PhD project. So we wanted to be able to see, first of all, if our methods were, let's say, not exactly accurate is not the correct word. We wanted to see if what we were doing was in touch with reality. Let's just put it that way. So we performed some zeta potential measurements and calculations on nucleosomes. To our knowledge, it is the first time that zeta potential has been used on nucleosomes. And I should say that in this, probably everybody knows, but with this technique, the mean value of the electrokinetic potential is measured by light scattering, and it is widely used in colloids or for liposomes. And so it allowed us to measure the electrostatic potential on the slipping plane. And then we also performed calculations of using Delphi to see if we were able to get a match, and luckily we were. So as you can see, usually with Delphi, we are overestimating the potential with respect to the experimental data. We think that this is because of the fact that we estimated the position of the slipping plane and using this formula. So we basically estimated it taking the physiological ionic concentration as reference, and then we use this to estimate its position for different ionic concentrations. And but we were overall happy because we even though we overestimated it, usually we were able to get a match nonetheless. So finally, I'm going to leave you with some conclusions. Chromatin is a multi-scale system in both space and time, and its study definitely requires the synergy of multiple modeling paradigms and experimental data. And aerostatics is of paramount importance in chromatin and for determining the topology of chromatin folding. In my project, we studied intranucleosome interactions by developing a methodology to investigate the aerostatic determinants of protein nucleic acid interactions. We also looked at, using this methodology, the effect of the histone tails, and we saw that they stabilize nucleosomes, they tune transcription, and we were able to see what happens when they are truncated. We were able to see the role of specific residues that were very interesting. And then we also looked at interactions between nucleosome, inter-nucleosome aerostatic interactions. We created a map of aerostatic interaction energies. And these calculations, they provide the basis for the parameterization of an analytical model and a coarse-grained, then custom force field. Finally, we looked at what happens with solvation in chromatin. So I really like this sentence, the nucleosome is a sponge. It is from a paper that I have cited here. Oops. Yeah, it is from this paper, which is great, and it's a very nice paper. So we saw that nucleosome is basically as a sponge. It is very highly solvent accessible, which therefore means that absolutely we should take into account solvation in the systems, and nucleosomes should be modeled as rigid spheres with a lot of care and a lot of attention to that. And finally, we want to place our study of aerostatics in the wider context of the custom coarse-grained approaches for chromatin. So that's that. And finally, I just want to thank, first of all, I want to thank Walter for inviting me and for being my supervisor. I want to help Professor Alberto Di Aspro, who is also my supervisor, and Dr. Silvia Dante as well, and the nanoscopy group and the concept lab, the IIT. And I have to say this. So I'm finishing, as I said, my PhD, and my defense is in a month and a half, actually. And I'm looking for a postdoc in Europe now. So if you're interested, you know when to find me. And if you know somebody who's interested, well, let them know. And yeah, thanks for your attention.
Thanks, Artemi. So I hope this will be an encouragement for other young colleagues to make their presentation. And so I see that Marilyn wants to, this was just, okay, thank you.
I do have a question if I can.
Okay.
That was great, but it was also, you know, just very, very nice, very clear. So these are really highly, and I think coarse graining is clearly, you know, ambitious and creative way to get things done. So, but these are molecules with just a huge number of charges, and I'm wondering, like, what you do for, you know, all the lysines charged, or all the, you know, do you have a lot of ions to, you know, counterbalance the charges? How are you handling that in this model?
Okay. So you mean what's going to happen, the coarse graining, or?
Well, at any level you want to tell me, at the atomic level or at the coarse grain level. I mean, what are you doing with, you know, or is everything have canonical solution charge? Are you adding a lot of ions? Are you using, you know, so what, you know, what's your just general strategy for something these highly charged?
Well, it depends. It depends on what I want to do. So it's not in the presentation, but we've done also full atom molecular dynamics with these beasts. And yeah, in that case, you just add a huge number of counter ions because you need to balance the charge. Like we did a simulation where we had, in order to look at what happens to the torsion angle between two nucleosomes, we did a simulation where we have two nucleosomes connected by linker DNA in explicit solvent with everything atomic, full atom. And yeah, there you just add ions. You can, when it comes to Delphi, that's a different approach because it's a continuum. So when I do calculations with Delphi, I just ask it to have physiological ionic concentration, and that's it. I guess it really depends on what you want to do. When it comes to the analytical model, I guess it's, I mean, I don't think we do something about that in that case. I mean, it's not, I'm not the one developing it. So yeah, I'm just thinking about
Okay, well,
yeah, okay. But yeah, so also when it comes to the what is going to be the simulations, the molecular dynamics when we have the ambitious force field, then I guess it will also depend on exactly what kind of phenomenon you want to look at. But yeah, it's difficult to, sometimes they're difficult to treat exactly because they're very highly charged. So also with Delphi, when it comes to convergence, you need to be very careful that everything is, you know, kosher and stuff. So yeah, I hope that answers it to some degree.
yeah, I just want to make a comment. I mean, you might, you know, and I don't want to take too long. I mean, you might just see whether all the lysine or all the protein side chains are calculated to be protonated, you know, or whether they're turning themselves, whether some fraction of them are turned off. That would actually be quite interesting to some.
That's actually something that we're working on right now. Like the protonation state of nucleosome is something that, because it's going to be part of work that we're just finalizing now. So yeah, that's a very timely question.
So I think Alexei wants to make a question.
Oh dear, okay.
Yes. Well, first of all, thank you. Very nice talk. I really enjoyed it. Lots of interesting stuff. So I hope that to see a lot of it in press. So I have a question and suggestion, a comment on Marilyn's comment about protonation states. Okay. So the question, I'll start with the question. So you mentioned in the beginning that so you were surprised to see that actually the C-terminal, the small pieces, they play a role in the overall stability of the system. Yeah. That's really interesting. So I was wondering if you try to quantify that. And what I'm driving at is the following. So there was so experimentally a while back, Gottesfeld and Luger, they measured the free energy cost of cutting the tails completely. So how much the nucleosome is destabilized if you cut the tails. And the answer is about 1 to 2 kcal per mole, and this is the whole tail. So I wonder if you looked into the contribution of C-terminal, sort of way of connecting with reality of the C-terminals, or if you haven't, that may be a suggestion for the future.
Yeah, no, I didn't look at, so this work, I didn't say it before, but it's been published in Computational Structural Biotechnology, and it was published this year. But I didn't look at the free energy cost, let's say, of cutting the tails. What I did in this work was mainly I looked at how the changes in the position were leading to changes that I saw in the signal of the field and of the force. And then I tried to understand how this was connected to even slight changes of, like, slight shifts, or sometimes the residue might have been just slightly closer to the DNA. And yeah, what I saw for the C-terminal, for example, is that it might just move by a couple of angstroms, let's say a bit more, like 5 angstroms, and I would get like a massive like just massive signal changes in the electrostatic field. So, and then I started looking at what the literature was on C-terminal tails, and usually they were just, they were just ignored. So I was like stricken by that.
Yeah, that's very interesting.
Yeah. But it's not, I didn't really quantify it, like saying, for example, that I have, I don't know, a change of 5 times the field or something like that. It's not that, it's more a qualitative description. It's not that.
Right. So you could potentially, I mean, of course, the whole free energy would be difficult, the entropy and whatnot, but you could do the electrostatic part. Let's say you zero out the charges on those C terminals and just how much they contribute just electrostatically by comparison with, say, N terminals, and then compare with experimental how much of it. So you could still do potentially some quantitative analysis within the level of the theory you're working with. That could potentially be interesting. So one more suggestion I have is that very interesting work on when you pull away the nucleosomes and see different in the energy cost of different conformations. My suggestion is to look at, there's a work from Korolov and Lars Nordenskjöld where they looked at various mutual orientation. They analyzed PDB, various mutual orientations of the nucleosome. Again, it would be interesting to see to map those ones that naturally occur in various structures to the energy. So again, mutual electrostatic energies, like what is the energy cost? And finally, this is just a comment. Finally, comment on what Marilyn commented. We have done this analysis when we looked into the paper from 2018, when we looked into free energy of various post-translational modifications in the nucleosomal core. We actually did, it's in the appendix, we did the massive calculation of protonation states in the nucleosome. And it is very interesting. So of course, as you expect, this thing is highly charged, but there's a lot of really strong shifts in, let's say you have glutamic acids or aspartic acids with all over the place. You have gigantic pK shifts, and of course that you expect, right? It's a highly, you can have strong burial somewhere. Some sites are strongly buried, some sites are strongly buried next to the DNA. So we haven't looked into, detailed what goes where, but there's lots of shifts, and we've, some of it is in that paper.
Oh, well, that's great, because that means I don't have to do it myself. So thanks.
Well, we didn't look into details, but this is something that.
No, that's great. I didn't know about that. So thanks for pointing it out.
Yeah, look it up. First author is Andrew Fenley. It came out in 2018. In the appendix, there should be this, because we try to take the protonation state change into account in the calculation of the free energy. That's why we did it.
Cool.
There is a question from Thomas Lauer.
Hi, I just wanted to point out that the Stokes radius is your slip plane, and that's a readily measured parameter with great accuracy. And I would be stunned if Ken Van Holde or Jeff Hansen hasn't already made those measurements. So you could actually put a measured value in there. And secondly, you could measure the charge in your particles quite readily by dynamic light scattering, electrophoretic dynamic light scattering, and then compare back what you measure with what you calculated.
Okay, thank you. Oh, we actually were not aware of that. We looked into it, but we didn't come across it. So thanks for pointing it out.